Okay, well, welcome to this innovation showcase. We've had a really fantastic roundup of companies presenting, and today we are joined by, well, we've got a pre-recording from AviadoBio, who unfortunately weren't able to make it today. We've got ImaginAb, and we've also got the Quadram Institute. So just a few logistics before we get going. If you don't mind keeping your microphones on mute throughout the presentations, that would be great, but please feel free to use the chat box if you have any questions or comments. Both of our presenters today will actually be at Genesis tomorrow, so feel free to hold on to those questions and you can try and grab them on the day. And as you may have already noticed, we are recording this session. So that is it from me. I'm going to see if I can now pull up the presentation from AviadoBio. So just bear with me one moment. Okay, can everybody see that okay? Brilliant.
Good morning or good afternoon. My name is Graham Fielder. I am the Chief Operating Officer of AviadoBio, which is a UK company focused on creating transformative medicines for neurodegenerative disorders. Now, we're a company that is a spin-out of King's College London from the laboratory of Professor Christopher Shaw, who is a neuroscientist and neurologist who has focused his career on identifying genes involved in neurological diseases, namely frontal temporal dementia and motor neuron disease, understanding their biology, creating animal and cellular models of these, and then also conducting clinical trials for new medicines coming through. And the company was the genesis of his career in a way, in that he's been working very, very hard on these disorders, but unfortunately not a lot of progress has been made in terms of actually drugs that benefit patients' lives. And so unfortunately he's starting to see the patients who are children of the parents that he once diagnosed several years ago coming through and feeling that he hasn't, or the field has not yet advanced in terms of these new therapies. And so he's dedicated the rest of his career to helping meet the promise that he made to his patients, to help bring through some new therapies that can have transformative impacts on them. And so the company started in the laboratory down at King's College, and Chris and the team started to make a number of therapies that could be targeting these disorders. And the company is focused on creating AAV gene therapy specifically, so adeno-associated viruses, which are non-disease causing viruses, to help us deliver genetic material into the central nervous system to help address some of these diseases. And we're focusing on neurological or neurodegeneration for obvious reasons there. It's the largest cause of disability worldwide and the second leading cause of mortality worldwide. And so these viruses, we can use them to deliver different types of genetic material to, one, either supplement a functional copy of a gene that's mutated in a patient, or to simply add a new protein that could have some downstream therapeutic benefit. And then secondly, a lot of neurological diseases are caused by the toxic buildup of proteins or RNAs. And so we can deliver some genetic material to help the degradation of those toxic compounds as well. But one of the key things we're focused on is on delivery of these genetic medicines, the CNS, both the brain and the spinal cord. And this is because delivery, we believe, is the biggest challenge in the field. Diseases of the brain and spinal cord typically start focally, but the vast majority of them do not stay focal. They spread throughout the whole brain. And so it's very important to be able to distribute the drug as far through the brain you need it to the specific regions that are actually affected in the disease. And getting things into the brain is not hard. It's not easy as well. The brain and the spinal cord are actually built to keep out things from safety mechanisms. So the blood-brain barrier is the common one that people will point to. Very, very hard to get things into the brain. And this includes the current routes of administration that we use, whether it's into the periphery or into the cerebral spinal fluid. There's the current routes that we use to get into the brain and spinal cord do not give us sufficient distribution of these drugs to really have a transformative impact in these patients. And so that's kind of one of the main things we're trying to address in the company. And so I guess how this comes all together is I typically talk about it in 3 different topics or facets coming together. One is just we're now in a new generation of genetic medicines or gene therapy, and there's newer ways that we can think about creating these gene therapy vectors that are a little bit more specific, a little bit more precise, and responsive to the disease environment, that enable them to be not only potentially more efficacious but safer as well. Combining this with what we know about the diseases, the disease biology, the neuroscience that underpins them, and then how the neuroanatomy is linked to them is really important. And then this third piece, which is probably the most critical for what we're focusing on, is delivery routes, delivery technologies to make sure that the gene therapy we're targeting gets to the disease specifically in the specific regions of the brain or spinal cord that really matter. This is probably most accurately demonstrated in our lead program, which is delivering these gene therapy vectors to the thalamus. Now, why the thalamus? Well, the thalamus is the central relay station of the brain. There are billions of neurons that are within the thalamus itself, but they actually project to many more billions of synapses around the way. And so rather than the underlying neuroanatomy of the brain and spinal cord being the thing that challenges and prevents things getting into the CNS, how can we utilize the existing architecture there to help us deliver our drug rather than impede it? And so what we have found in our research is that by delivering small amounts of viral vector to the right places in the brain, we can utilize the underlying neural networks to help us deliver the virus, the RNA and protein to the places that it is needed to go, and quite substantially. And this is kind of where the name Aviado comes into. Aviado is Esperanto for aviation or aviator. So we typically talk about ourselves as navigators of the brain. How do we direct the drug to where it needs to go and not where it doesn't? So this is our pipeline. We focus on FTD and ALS right now. We have broader ambitions across neurology, but there is so much unmet medical need within these indications, and we have gene therapies to target as much of it as possible. And so our lead gene therapy is in frontal tubular dementia, and specifically a genetic subtype where patients have the progranulin gene mutated. These patients typically have between 30 and 50% of normal levels of the protein. And it's actually the second most common early form of dementia, second to Alzheimer's. And this is where adults, generally in their late 40s or early 50s, develop a progressive and quite rapid decline in personality, behavior, executive function, and or language. And unfortunately, it leads to their death in around about 7 years' time. So very aggressive, quite different from Parkinson's or Alzheimer's in terms of the progression. Unfortunately, these patients, there is nothing available to them in terms of treating the disease, only symptomatic management. And this gene that is involved, it's where patients have one mutated copy, and so hence 30 to 50% of the normal level is present. And this gene, progranulin, is involved in sort of the waste removal system of the cell. And so without it, the neurons don't function properly, causing their death. So we have developed a gene therapy to be a one-time treatment that replaces a new functional copy of the progranulin gene into the brain, and specifically neurons, to enable the brain to start producing its own normal level of progranulin again. And importantly, from a safety point of view, it has been designed such that it is only active in the brain and not where it is not needed. And as I mentioned earlier, the approach here is to deliver the virus that has the functional copy to the thalamus of the brain in a minimally invasive surgical procedure to then allow the thalamus and the projections from the thalamus to deliver the gene therapy around the brain, and particularly the cortical regions of the brain where which are mainly affected in this disease. Now, we've been undertaking extensive non-clinical studies in a number of species, particularly the disease mouse model, which presents with a similar pathology as patients do. And what we can show is both we can suppress and ameliorate the pathology that is found in these mice by giving them small amounts of the gene therapy to initiate and raise progranulin levels in these mouse models. We've also used some larger animal models. These are non-disease models, but the wild type animals, to really understand safety and distribution of these therapies. We like to use the sheep because there's a really, really large brain compared to a lot of other species. It's around about twice the size, at least, of a macaque or an HP. And so we've used the sheep extensively to understand how we can use these small doses and where do we put it in the brain in order for it to get this really profound distribution across the brain. And then NHPs, as the preferred toxicology species, we have been well characterizing the drug's biodistribution, but also safety in those. And we're coming to the completion of all these studies as we speak, such that we've already begun to submit clinical trial applications into a number of geographies, including the UK, Europe, and US, with intentions of starting a clinical trial as soon as possible. And so here's just a brief design outline of the trial. It's called Aspire FTD. We were seeking to enroll progranulin deficient FTD patients into a phase 1/2 trial, where the primary objective here is obviously to look at safety of the gene therapy, but it's also an opportunity to understand some preliminary efficacy around some unique biomarkers that we have in this disease that help us understand if the drug is having an impact on neuronal health or inflammation within the brain, but also some key endpoints around imaging, around atrophy. And then within this disease, we have some very useful dementia rating scales that help us understand how the patient is doing across a number of different functional tasks. And so, as I mentioned, this trial is almost ready to go. We're hoping to have initiate the trial by the end of the year, with data starting to be read out in 2023. Beyond this lead program, we are focusing on a number of other preclinical gene therapies that are coming through for different subtypes of ALS, or motor neuron disease, or frontal temporal dementia. And we've focused on motor neuron disease, or ALS, because also obvious reasons as well. It's a rare disease, but it is actually quite common in that 1 in 300 men in the UK will have this on their certificate. And it's a very, very aggressive motor neuron disease where patients in sort of the prime of their life are diagnosed and rapidly progress to death within just a few years. And so we're bringing through a collection of preclinical programs, hoping to be in the clinic soon to hopefully address both genetic subtypes and more sporadic types of this disease. And of course, underpinning all of this effort is an amazing team. We're a team of just over 55, headquartered in London, with also some employees based in the US as well. We've raised just over $96 million US, and have been able to attract a great syndicate of very supportive investors, all of whom had significant experience when it comes to gene therapy or cell and gene therapy. And so we're very, very happy to have this really supportive group of investors to help make this vision of creating these transformative gene therapies a reality. Thank you for your time, and I hope you enjoyed my talk. Thank you.
Right, well, thank you very much for listening to that presentation. It was great to have a bit of an insight about what AviadoBio have been up to. Up next, we have got Mandeep. from ImaginAb. I'm just going
Just showing my screen, Jasmine.
perfect. Your camera's off at the moment as well.
Can everyone see it?
Yeah, that looks good. It's just in handout.
There we go. Is that okay?
Yeah, perfect.
Right, excellent. So thank you for the opportunity to speak. It's great to be a part of Genesis 2022, an innovation workshop, and delighted to be sharing more information about ImaginAb and our proprietary CD8 immunopet technology, and how this can see the bigger picture using whole body imaging of CD8 T cells for immunotherapy clinical development. And also really excited that we are one of the nominees for the Bio News Round Award. So hopefully this will inspire you to vote. So ImaginAb at a glance. We are a clinical stage revenue generating biotech company. We're headquartered in the UK and also the USA. We are focused on the rapid to market development of novel clinical imaging agents, in particular CD8 immunopet and also radiotherapeutics. And our imaging agents are underpinned by a proprietary antibody fragment technology platform, which has, and we have broad IP estate covering biological agents, our mini-body composition of matter, and also IP to specific targets, which cover the diagnostic and therapeutic aspects of those. We have a great experienced management team who have a strong background in life science and healthcare industries, and we're backed by quite well-recognized venture investors. So as I mentioned, we have two arms, next generation imaging and next generation therapy. So the imaging is specifically around immunopet and the therapy is around radiopharmaceutical therapy. And this talk will be focused on our immunopet work. In particular, ImaginAb actually lead the field in this space with regards to CD8 T cell imaging. We have the only CD8 T cell imaging agent currently in phase 2 clinical development, and we're on track for a market FDA authorization, which is expected in 2025. So we have a proven track record of developing radiopharmaceutical agents, and our winning combination is essentially our mini-body binders. And these are paired with a chelation system for biologics. So we have a chelator that allows binding of a radioisotope, and that allows imaging and also therapeutic functionality. And that is then has been validated, and validation is ongoing for that combination. So in terms of the technology, the fragmented antibody technology is proprietary. It specifically binds to targeted cells and delivers a radioactive payload in a very highly selective format. In terms of being clinically validated, that's currently ongoing with our CD8 specific mini-body. And we have the potential to deliver therapeutics with optimal efficacy and safety profile. So in terms of CD8 immunopet and what is it, how does it work with imaging? So our actual technology is called crofelemer budoxam. It is a mini-body that is radio-labeled to zirconium-89. Zirconium-89 is an isotope and it has a half-life of roughly 3.5 days. Overall, once it's combined, it has a product shelf life of approximately 6 days. It's immunologically inert because it doesn't have the Fc portion. It's technically half the size of a typical antibody and has a very high affinity for CD8. It also, because of the size of the mini-body, has quite a quick blood clearance and can be cleared from the system within 24 hours. This combination lends itself to a very high signal to noise ratio, which you can see in the image on the right-hand side. So the extreme right, the image there is a current standard of care imaging for most oncology clinical studies. And it's actually FDG PET, and it tracks the metabolic activity in the body, and you can see that represented by brain and bladder. And you can also see the arrow pointing to the tumor there in the leg. Conversely, we've got CD8 immunopet. You can see the resolution is slightly different. There is binding to normal immune tissues, so you have lymph nodes, spleen and bone marrow, but you can also see the detection of a tumor there. And what's interesting is it's not just a very dark blob, you can see different intensities of what's being detected. So as mentioned, we are currently in clinical validation for our CD8 immunoPET imaging agent. It's completed phase 1 and phase 2A. Phase 2A was actually presented recently at SITC, Society of Immunotherapy, and this was mapping the correlation between IHC, immunohistochemistry with biopsies, which is typically the routine procedure used to analyze CD8 T cells in tissue, and mapping that to imaging. So why CD8 immunoPET, and why do we think it's got value in immunotherapy monitoring? So we believe it's a novel disruptive technology. It's non-invasive, it's molecular imaging, and it offers an advanced monitoring of immunotherapy treatments and beyond. It has high sensitivity and specificity. It enables higher accuracy and potential to allow early detection of immune response compared to the standard of care, which is measuring the size of the tumor after a certain time frame post-treatment. It also offers the potential guide to biopsy. It can also offer the potential to replace biopsy frequency, and this is quite valuable given that a lot of these people are quite ill. It's painful to get, biopsy of a tumor, especially if it's in a hard to reach location. So we believe it has great value there. And also allows you to monitor systemic immune responses as a result of treatment and adverse effects. As mentioned before, the iCorrelate study, which is the phase 2A, is a quantitative measure correlating for biopsy for complementary monitoring disease progression and response to therapy, and that data has been presented. That study is complete. Also allows observation of heterogeneity of immune status and response in the body. How does this translate to the patient? How does it translate to immunotherapy monitoring? We believe it has two arms. So one is to accelerate treatment monitoring. So it has the potential to detect drug response earlier, and I can show some examples of that. Speed up trials through smaller focused cohorts to demonstrate proof of mechanism, and also determine if immunotherapy combinations are useful. So if you're combining your novel therapeutic, for example, with a standard immunotherapy such as pembrolizumab or a checkpoint inhibitor, we can look at using this technology, what the effect is with and without. And then monitoring disease progression, quantitative monitoring, enabling an early response measurement to standard of care. So in terms of getting that comprehensive whole body imaging status, this is just a visual to help you understand how this works. So how can a CD8 immunoPET whole body image help you show the bigger picture compared to a biopsy when looking at many tumors or metastases? So on the left side is an example of biopsy. It's one tumor, it's a little fragment or piece of tissue of that tumor. It's a microscopic view, it's invasive, and you're not always guaranteed that your tissue is going to be in the best quality, it's got to the right part of the tumor, etc. So there's a chance of failure. On the other hand, with the CD8 immunoPET technology, you get your whole body image. You're not just looking at one tumor, you're looking at many tumors across the body. Gives you that macroscopic view. It is non-invasive, so it's administered intravenously. It's repeatable, so we can do serial imaging, and you get to see the inter and intra-tumor heterogeneity, and I can show you some examples of that in the next slide. So I mentioned that using CD8 immunoPET can give you an earlier readout on a clinical response, and this is an example from our phase 2A study of what this might look like. So this particular these particular images are a demonstration of a patient who had melanoma. You can see it in the left leg there. And baseline is basically pre-treatment. So prior to treatment, this is what the patient's imaging status looks like. So we're comparing against FDG PET and CD8 PET. In the image below, what we have here is a slice of the leg. And then you can see it's actually a CT image, and we've overlaid the CD8 imaging on top. There's a measurement there called SUV max, which is standard uptake value, and that's really measuring the intensity of the CD8 uptake. So that's the brightness of the signal that you're getting. So at baseline, what we can see here is in the FDG PET, we can see the CD8 imaging overlaid. It's quite clumpy, homogeneous, not really giving too much detail other than that we can see it. However, with the CD8 immunopet, what we can see in more detail is almost like a ring-like effect around that tumor. We can see that there's a higher intensity on the outside and it's quite dark on the inside. And that might give us some indication as to, is this tumor immunosuppressed? T cells are not infiltrating this tumor. So it kind of gives us an immune status at baseline prior to treatment. On treatment, we're looking at 1 month post-treatment, so as a checkpoint inhibitor administered to this patient, and we're also looking at 4 months. So comparing CD8 immunopet with FDG PET. And what we can see after 1 month very clearly in this slice of the leg is that the SUV max increases substantially, and this is visualized through a higher intensity of CD8 readout. And you can see the brightness there, localized of CD8 around that tumor. And at 4 months, what this correlates to is actually a lower intensity of CD8 and a smaller size of the tumor. So this is really, really interesting. If we just looked at the CT images alone on the right-hand side here, if you look at the measurements of the tumor, so measuring the size of the tumor without looking at CD8 immunopet imaging, what we can see is there's not much change in the size of that tumor. But at 4 months, we do see a sizable change, almost 35% smaller at 4 months. And that's great, it's a partial response. But the value of CD8 immunopet imaging here is that actually at 1 month, you're getting an earlier readout, and it suggests that this patient is likely to respond to your immunotherapy treatment. So what we're saying is actually, if you were to apply the CD8 immunopet imaging earlier into your treatment cycles, what you can actually gain from that is an earlier response readout, at least 3 months earlier in this particular example. So in terms of classifying tumors based on CD8 T cells, this is another example of whole body imaging and what you can see looking at those individual tumors, so getting the micro and macroscopic view. This is one particular patient, renal cell carcinoma, and the images you're seeing here are all metastases. So you can see that we have imaging in the skull there. There's also imaging in the lungs. And what this imaging looks like is intense and homogeneous or heterogeneous. And it's just really giving you an insight into what that might look like in one particular whole body scan. And where the real value here is, if I draw your attention to the right-hand side, we can see a tumor here, and with the overlay of the CD8 imaging, you can see that it's not all one. You can see that there are brighter regions, less brighter regions. And what this indicates is that this is heterogeneous. There are hot and cold areas. So by hot and cold, we mean it's likely to be immunosuppressed or not immunosuppressed, and there is an infiltration of T cells in that tumor. And this correlates to a biopsy. We can see here in the biopsy of that region, there is CD8 T cells being detected in one part of that tumor and not in the other. And why we think this is valuable is if you were to do a biopsy alone, if you happen to just get into the immunosuppressed region, you would see no T cells, but actually the image tells a different story. There are areas of this tumor that do have CD8 T cells there, and this would give you a starting point prior to treatment, and you can see how that tumor would respond post-treatment. So how are pharma using CD8 imaging today? So it's typically 2 scans. There's a baseline scan and there's an on-treatment scan. The purpose of that baseline scan is before immunotherapy is administered. It would allow you to define the CD8 status in tumor microenvironment. You could even stratify your patients based on their immune status and also guide intervention to biopsy. Post-treatment, it allows you to confirm the mechanism of action. Is my drug working? How, what dose is it working at? Can it predict a drug response? So look at the efficacy earlier than standard of care imaging. Allow you also to prioritize tumor types and stratify patients. In terms of the validation adoption of CD8 immunopet, we've had some ongoing collaborations and partners since 2017. We actually formed a collaborative group with Pfizer, Takeda, and AstraZeneca in 2019, and we also have a partnership with Roche. We've had ongoing partners since that time, and as you can see, 2022 is quite an active year for ImaginAb. We have application of CD8 immunopet in a range of therapeutic classes, such as T-cell engagers, cancer vaccines, immunomodulators, autologous cell therapy, oncolytic viruses, and so on. And we have also had some studies in COVID-19 subjects too. And in terms of our partner Roche, I wanted to use this opportunity to also highlight that they are presenting some clinical data where CD8 immunopet was used in some imaging in a trial using glofitamab, which is a CD3/CD20 bispecific T-cell engager. And this data will be presented at ASH, the American Society of Hematology, next week in a poster. You can actually view this poster on their abstract website, some elements of it. And so we're very excited to see that the application of our technology is being presented by our partners now. In terms of clinical development, I mentioned that we are on track for an FDA approval expected in 2025. And the purpose of this is to really define CD8 immunopet as predictive and prognostic value in immunotherapy clinical studies. And these studies, the clinical validation has been ongoing since 2017. 2021 and 2022 were very impactful years for us. We're able to show more use of CD8 immunopet as predictive and prognostic tool in certain studies. We have a network of over 30 validated PET sites, and patients are being imaged in the US, EU, and Australia. And we also have guaranteed manufacturing in 3 continents globally. Our phase 2b studies have been initiated. So we have the iPredict study ongoing, PRETZEL is just enrolled, and we also have a neoadjuvant study ongoing in the US. As I mentioned, it's being applied in diverse immunotherapy programs. And notably, the most exciting thing for us, and the purpose of this talk really, is also to highlight that we, in May 2022, we signed an exclusive license with Dongcheng Pharmaceutical Group in China, who will be commercializing our cancer radio tracers across Greater China. We're really excited about this simply because, you know, that in China, there's been a massive upward trend of clinical trials, and also the technology that is coming out of China at the moment, and notable companies such as BeiGene, CStone, Legend, Innovent, just to name a few, are getting quite a lot of traction, adoption, and many of these therapies are now also being validated in other countries around the world. So really exciting collaboration for us, and hopefully it will provide an acceleration with this informative data showing the efficacy and proof. So in summary, ImaginAb is a revenue-generating clinical stage biotech. We're generating over $26 million of revenue generated from existing pharma licenses. We have a novel specific antibody fragment technology platform, which has an optimal safety profile for diagnostics and therapy. We have a first-in-class clinical asset, and this allows whole body imaging of T cells, CD8 T cells, addressing an estimated market of between $600 to $1.1 million. Our supply chain for the radioisotope is guaranteed, and we will have no issues with that in terms of shipping that out globally. We have a robust IP and a fantastic management team and advisory board who have extensive clinical development and commercial life science expertise. If you wish to learn more about our technology, there is published data that you can access. There are clinical trials actually that you can find on ct.gov, notably Roche, HooKeeper, one of our partners, and also AstraZeneca. All of our investigator-led crefmelimab trials are visible and you can find that in ct.gov by searching for crefmelimab. And we have a number of other clinical publications and abstracts, which you can find on our website, so I encourage you to visit. You can also join our Knowledge Hub. And we also have some other valuable content on there, such as webinars, which help explain the application and significance of the technology in more detail. And that's it from me.
Thank you, Mandy. What an exciting year for ImaginAb. My goodness, you have been busy. And thank you for highlighting the BioNews Round Award. There is still time to vote, so in just a moment I will pop a link to that in the chat box. But yeah, thank you. Up next we have Roberto from the Quadram Institute. So I'm just going to bring you up, Roberto. There we go. I can see your slides.
Yep. Fantastic. You should be able to see the title slide now.
Yeah.
Fantastic. So my name is Roberto Zanchi. I'm the head of business development at Quadram Institute. So the Quadram Institute is a partnership between Quadram Institute Bioscience, who formerly was the Institute of Food Research, the University of East Anglia, and the BBSRC, and the Norfolk and Norwich University Hospital, which is the local hospital in Norwich. So as you can see, we are housed in a state-of-the-art new facility that was represented as £75 million investment by BBSRC, and is underpinned by over £40 million of investment in the science equipment and the capital equipment. So we are based in the Norwich Research Park, and this is a cluster of with world-leading institutions. So as you can see, we are just next door to the Earlham Institute, with whom we collaborate very closely. But also we're very close to John Innes Centre, as well as the University of East Anglia, the Sainsbury Laboratory, and the hospital. So this is a fantastic hub. And I'm just going to walk you through on this, on this slide, to give you a flavor of what our capabilities are and the research that happens within the building. So on the lower ground floor, we have our regional endoscopy unit. So this is one of the largest endoscopy units in Europe. It can process up to 40,000 patients a year, and it can be staffed to up to 100 clinicians. We also have a clinical research facility, which is on the ground floor, and that really links the clinical work that goes on in the regional endoscopy unit with the research laboratories, which are the top 2 floors, and they can house up to 300 scientists. And we also have part of the space within the building that has been reserved for collaborative innovation labs, which is to incubate spin-outs from our institute, as well as host strategic partners for the institute. So our mission is to delivering better, healthier lives through innovation in gut health, microbiology, and food. And we focus really on 2 areas of research, one aimed at promoting health and the other reducing threats. So we have a number of expertise represented across our scientists. We have a number of strands of research on food and personalized nutrition in particular, and how that food is digested in the gut and interacts with the microbiome, and how the metabolites are then deliver health benefits to not just in the gut area, but to the liver, and also more distantly, for instance, in the brain. So there are a number of axes that we are studying. On the other end of the spectrum, we're looking at, we have deep expertise in microbiology, and that allow us to investigate, the pathogen and how to reduce pathogen transmission in the food chain, as well as decrease the potential for the emergence of antimicrobial resistance, which is, a major problem that is limiting, the procedures that can happen in hospitals these days, and is a major health emergency that needs to be tackled. But we're also looking at how biofilms are formed, both in the terms of, for instance, in AMR and UTIs, but also the roles they play in food spoilage. And of course, between these 2 areas, there is a lot of overlap. And so we are also doing a lot of interdisciplinary research and applying it to real problems, and trying to deliver real outcome for patients. For example, we are setting up a fecal matter transplant facility that is going to be GMP certified and licensed under the MHRA, and that will aim first at treating patients with recurring C. diff infections. But in the medium term, we're also looking at using it to investigate how FMT can be used to treat and deliver outcome for patients suffering from other diseases such as Parkinson and ME. I'm going to give you a quick highlight of our capabilities that are available, in research collaboration with our partners. One important capabilities for the area we work in is bioinformatics. So we have built quite a bit of a capability in this space. Primarily, we are able to run both shotgun sequencing through our 4-luminous sequencer, as well as a long-read sequencing through the Oxon Nanopore platform. And we have, of course, also complemented that with bioinformatic capacity, both in terms of CPU power and data storage. So this allows us to analyze the metagenomes from the microbiome samples we collect. And also, we have developed standardized and validated pipelines to analyze this data. And we make that available through collaboration with our partners. But also, we are leading on the cloud infrastructure for microbial bioinformatics, CLIMB, which was funded by the MRC. And essentially, this is an instrument and is a platform that is available, that has facilitated academic research throughout the UK in this space. We also, as I mentioned, have access to a clinical research facility that is housed within our building. It's been particularly designed to perform both observational and interventional clinical trials in nutrition and microbiome interventions. To date, most of its actually use was in testing COVID vaccines, because our building was inaugurated just before the pandemic. But nowadays, we're also starting to use it to, for instance, monitor the blood glucose response to innovative ingredients that we have been developing in the Institute that are aimed to lower the digestibility of starch and therefore lower the glucose spike after eating a meal. So this is one of the examples. And also this facility underpins, all the sample collection that forms a resource for our scientists and for further collaboration with companies. As I mentioned, we also have a biorepository. This is in collaboration with the University of East Anglia and the Norfolk and Norwich University Hospital. And it facilitates a collection of tissues, both at the hospital and at the clinical research facility, that it can be used and is being consented for use in our collaboration and fundamental research. We also have a food data bank national capability. So we've been curating food composition for decades now. And this can support research and innovation, as well as allowing companies, and we can provide this as a service. We can provide nutritional composition and labels for foods. And finally, whilst we work primarily through collaboration, whether it be large partnerships or consortia, down to perhaps studentship, PhD studentship collaboration. We can also work on a fee-for-service base through our subsidiary QIB Extra. So we can provide bespoke scientific research, as well as a consultancy and particular services around the authentication of food and the analysis of food. So myself and the members of my team, Leila and Stephen, are going to be at Genesis tomorrow. So we would welcome to have a chat with anybody that has questions, and we're always open for collaborations. So we look forward to meet you there.
Brilliant. Thank you so much, Roberto. Do we have any questions or comments before we round up in the chat box for either Mandy or Roberto? I mean, as we said before, if you've got any questions that you would like to ask them in person, please do so tomorrow in London. Wait just a moment just to see if we have any questions come through. No. Okay, wonderful. And we do have, so I did pop the link in the chat. If you haven't voted already, make sure you go and do that now. And our next session will be on Thursday now, Thursday morning. So we hope to see you there. And thank you everybody for joining today. Thank you again to our presenters, and we look forward to seeing you tomorrow. Thank you.